Quinto Congreso Internacional en Investigación e Innovación en Ingeniería, Ciencia y Tecnología de los Alimentos, IICTA 2021, modalidad virtual. Sistemas alimentarios inteligentes, sostenibles y saludables. Octubre 13 al 15 de 2021, organizado por la Universidad Nacional de Colombia, sede Manizales. Con más de 500 participantes, expertos nacionales e internacionales. Intercambio de conocimiento entre la comunidad académica y empresarial del sector agroalimentario. Exhiba sus investigaciones y productos. Sistemas alimentarios sostenibles. Se tratarán 11 temáticas. Estudio sobre efecto del COVID en la cadena de valor de los alimentos. Economía circular, sostenibilidad y seguridad en el sector alimentario, entre otros. Conferencias y cursos pre-congreso, premios Larkin. Para más información visita www.icta.manizales.unal.edu.co. Quinto Congreso Internacional en Investigación e Innovación en Ingeniería, Ciencia y Tecnología de los Alimentos, IICTA 2021, modalidad virtual. Sistemas alimentarios inteligentes, sostenibles y saludables. Octubre 13 al 15 de 2021, organizado por la Universidad Nacional de Colombia, sede Manizales. Con más de 500 participantes, expertos nacionales e internacionales. Intercambio de conocimiento entre la comunidad académica y empresarial del sector agroalimentario. Exhiba sus investigaciones y productos. Sistemas alimentarios sostenibles. Se tratarán 11 temáticas. Estudio sobre efecto del COVID en la cadena de valor de los alimentos. Economía circular, sostenibilidad y seguridad en el sector alimentario, entre otros. Conferencias y cursos pre-congreso, premios Larkin. Para más información visita www.icta.manizales.unal.edu.co.
Good morning. Welcome everyone to our Cycle Pre-Congress Conference to the FI International Congress on Research and Innovation in Engineering, Science and Food Technology, IICTA 2021. Today, we have invited to the Professor Yixiang Xu. She is an engineer, master, and Ph.D. in food science and technology. She has more than 20 years of experience as research and professor. She is a professor at the Virginia State University. She is editorial board member of Journal of Food Processing and Technology, and she has more than 35 international publications. The participants can write the question in the chat box, and when she finishes, the professor will answer their questions. We welcome to Professor with her conference called Processing Technology for Development on Plant-Based Protein Products. Welcome.
Good morning. Everyone can hear me?
Yes, Professor, I can hear.
Okay. Hola, good morning. Thank you for introduction. And my name is Yixiang Xu. And so just call me Sue. I'm the professor of food processing engineering at Virginia State University. And today I'm going to talk about processing technologies for development of plant-based protein products. And first, before starting, I want to briefly talk about myself, my education background, and so my career background. Original, I'm from China, and so my hometown is called Urumqi. Can you see it? I just want to make sure. Can you see the screen? Hello? And so I went to Southwest Agriculture University for my bachelor and the master. After I finished my master, I went to Qingdao for my Ph.D. For working for 5 years at the Ocean University of China, working on the seafood processing and technologies. So in 2001, I come to United States and at University of Nebraska-Lincoln doing for my PhD. And
sorry, Professor, can you put the in mode presentation?
Oh, I'm sorry. Can you see it?
No, in this moment, don't see the presentation. I see your screen. Oh. Yes, and in the down part of the left down part, you put the presentation in the PowerPoint.
Okay, just okay. Sorry for that, because this computer is still need a share screen.
Don't worry.
It's okay? Can you see it right now?
Professor, in the down left, you put the presentation. You see a draw to computer or to similar.
Yeah, so I put the share. Can you see it right now?
Yes, I continue. You can continue.
Okay. Yeah, and so I just introduce myself. So my name is Yixiang Xu. I'm the professor at Virginia State University. Before I come to US, actually I was originally born in China, and so my hometown is called in the southwest of China called Urumqi, Xinjiang. And then so I went to the place called Chongqing to do my bachelor and master in the food science and food engineering. So after my education, I went to Qingdao in the Ocean City for the work for 5 years in the Ocean University of China, mainly work on the seafood processing and the technologies. And in 2001, and I come to United States study in the University of Nebraska in the food science and the technologies. And so after finish my PhD in 2005, and I did a postdoc and a research assistant professor at the same institute. Then so in the 2010, I got a job offer in the Virginia State University. And so I've been here for 11 years. So rising from assistant professor to professor. Regarding for Virginia State University, so I'm actually in the College of Agriculture. For College of Agriculture, we have 3 components, teaching, research, and extension. For research part, we work on the plant science, animal science, and the food science. So I'm in the food science area. So my research program actually is food processing and engineering. So I'm the PI, I'm leading the program, but supported by my research specialist and graduate and undergraduate students. For my research interest and mainly as a food engineering, so my main interest is including 4 areas. One is sustainable food processing and packaging technologies to improve food quality and safety. And we also I'm interested in value added utilization of agriculture products and biomass. And also right now, mainly my project, we're working on development of plant-based protein products, which is the main topic today. I'm going to talk about it. The last topic, I'm also working on the nanotechnology in food processing and packaging. That's my research interest. For my research projects in the last 10 years, my research project, so we got a different funding from federal government, like United States Department of Agriculture, also a local agriculture agency, and also like the industry and for other institute. So research projects mainly focus on is one is active biodegradable nanocomposite films for food packaging. So for this project, and it's implement sustainable food packaging ideas because we use biodegradable polymers, which comes filled and meaningful starch. Then we make nanocomposite. So use a nanocellulose crystal. So nanocellulose crystal meaning from like grass and wood has a cellulose. In the meantime, we're also working on the active packaging. So in active packaging means you have some active compounds adding in packaging. So we use the grape humus extract. Actually, grape humus is a residue after grape press the juice, so left skin and seed. Actually is a low value. So what our purpose is try to extract the antioxidant and antimicrobials. Then we put in the films then to extend shelf life. That is one way we did a sustainable food packaging area. And another part, so which the project we're still working on is we use non-thermal technologies to reduce the meat in processing meat, reducing salt in the processing meat, because in the Western diet, especially in the US, salt is very like is extensive, high for some food, especially in the meat. So what we want to do is using some non-thermal technologies also function ingredients to replace the salt function, therefore to can reduce the salt. So this project has been funded by USDA, so we're still working on that. And so we're also working on the plant-based products. And so meaning is funded out for different protein source for the we want to understand the protein and the legumes, their processing, nutrition, and functional qualities. So today, and so my main talk going to talk about plant-based products. So plant-based products right now become very popular, and because the trend for consumer, they're looking for healthy food, and so another one is because plant being found is more sustainable compared to raised animal, because the raised animal need more land, water, and resources, but plant going to get has a less requirement. So that's why, because also people care about health. That's why right now plant-based food has been increased very quickly in recent years, especially in 2020. U.S. sell plant-based products increased 27% to $7 billion. So this trend is going to be continue. Expected by 2027, they're going to reach 74.2 billion, the plant-based products. Of the plant-based products, protein is a core of the ingredients in the market. Currently, and the main plant-based products on the market, one is plant-based meat, another is plant-based dairy products. For plant-based meat, meaning it's for you say for the Beyond Meat, Impossible Meat. Right now, if you go to like a Burger King, you go to grocery store, is already a lot of plant-based meat available. Even a lot of meat company like Tyson, they also put investment on the plant-based meat. And so that's the topic become a really hot topic. And so meaning for plant-based products, and the source, people use soy protein. But soy protein has an issue because the allergy, also the other the issues. So people looking for other new novel plant protein source. Actually, in the plant-based meat, right now, most successful the protein used is a pea protein. And for us, so we working on the chickpea. And so we've been working on the chickpea about, since I joined here about 10 years. The background is because we are located in the Virginia, and so about 2 hours south of Washington, D.C., and has one famous company called Sabra. And Sabra actually is a hummus company, and the original located in the New York. In the 2010, the company relocated to the Richmond, and Virginia is about 5 miles from our university. So we started beginning to collaborate with the company to understand the chickpea and their protein or the potential application, and how the science behind, and when you cook chickpea, how each ingredients in the chickpea, and they can be used as functional ingredients. So today, my mainly talk about our efforts or study working on chickpea and their protein. I want to mainly talk about how we processing, how the nutritional and the functional properties for our research on this area. chickpea actually is also called ganzazu beans. Actually, it's in the world, it's the third most important pulse in the world. And I'm sure, and in the Latin American, you have a lot of chickpea too, because mainly in the U.S. It's in the north part, like California, also in the like South Dakota, Wyoming area. And so chickpea has a good nutrition content. They have a lot of essential and total amino acids and the macro and the macro minerals. Also is a healthy food. So that's why they use a lot of, many use for like the hummus, which is the Sabra hummus. We work with them, and it's the #1 in the United States for the plant-based dips right now. They also can use other products like soap and salads. So our project, when we start this project, actually, and because we're working with industry, we want to understand when the chickpea being harvested and going in the different location areas around the world, we want to understand their physical, chemical, and function properties of the raw chickpeas. Then after this, we want to understand how these properties, like nutritional, functional, structural properties, affected by different processing methods. And for example, like a pressure cook, microwave cook, or just soaking. Then so because as legumes, and the chickpea has a high protein content. So our next step is we want to isolate protein from raw and even for processed chickpeas. We want to characterize their properties, especially for processing chickpea. We want to know after being processed, and we want to see how the chickpea, the protein changed during this cooked process. So initially what we did it, and we just compare like soaked, unsoaked chickpeas. And after soaked and unsoaked, we did like some did moisture heating, and such as pressure cooked or microwave cooked, or even dry heating is roasted, because like a lot of area I know they eating like roasted the chickpeas. So after we conduct processing, next step we did is characterization. We measure basically including like physical properties, including like seed weight and volume, total solid lost, bulk density, hydration, swelling capacity. So we also measure like nutritional or chemical composition, such as like protein, oil, ash, total carbohydrate, total starch, resistant starch, fiber, amino acid, and anti-nutrients. Because as food ingredients, even they are very nutrients, but also important is how they behave when they put in the products. That's why we also measure their function properties. Function property including like oil, water oil holding capacity, emulsify activity, stability, forming capacity, and the protein solubility, in vitro protein digestibility. So our job is we have the raw chickpea. We also have cooked chickpeas. So we want to know how this different cooking process affect their different properties. So I'm just a brief talk about what our finding, and also have some publication. I'm going to list the publication. If anyone interested about the details results and finding for our papers. So based on our research, we found like a cooking process could increase fiber, total carbohydrate, total and resistant starch, and also amino acid profile and in vitro protein digestibility. But they decrease like mineral content, protein solubility. And so these two is the nutrients we want to have more, but cooking reduce it. Of course, it's good because reduce the tannin and the fatty concentration. These two actually is anti-nutrients. Cooking process reduce anti-nutrients, which is a good thing. And also, cooking reduced emulsify activity, stability, and forming capacity, which means their function properties reduced. With all different cooking methods, we found soaking with microwave cooking probably the best method for improving nutritional qualities. This work do for the whole chickpeas. We still have like a protein, carbohydrate, and minerals. So based on the research, we published like 2 papers, and in the 2014 and the 2016, anyone if you're interested for the details of our findings, and so please send me email or just send in the box so I can send the paper to you so get more idea. So for what we're working on that. And after we finish work, actually is another one is one people, and actually is based on Italy. And so they working on the healthy and good cooking method. Then they found papers. Then they put their own website and to see like post it to say is it better to microwave or pressure cook on the chickpeas. And because this is a group, they're interested in what kind of processing make the good for the food. So next step, after we did for the chickpea the whole, and we go the deep, we just focus on the chickpea the protein. Like I mentioned, and so because chickpea protein is one of plant-based protein. Right now has a trend to increase in the plant-based diet, especially for legume. Legume is really hard protein source. Like I mentioned before, like pea protein right now has a lentil protein, also this chickpea proteins. And the chickpea protein, usually chickpea has about 20% protein content. They have has a good amino acid balance. And so protein profile is really easy, highly by availability, easy to be digested. Is a good protein source to use. So what we did it as all other protein, we isolate the protein, use a precipitation method. Basically, we just define our protein as a chickpea powder. We ground the chickpea become the powder. Then so we define it, use a hexane. Then so we just disperse in distilled water, adjust the pH to 8.5 and to soluble or protein. And then become a soluble protein. Then we adjust the pH to 4.5 to precipitate protein. And then so we use the water, wash with the water, and to remove any salt. Then we freeze dry it before we contact all different character their properties. So what we did it, and we measure, like look at their color, and we look at their functional properties, and also their like secondary structure, like alpha helix, beta sheet. We also look at their protein profile and the thermal properties of chickpea protein. here is some of the results and for our funding for the because what we did we use the raw chickpea, therefore extract the protein. In the meantime, we also use different cooking methods, like we soaked, we roasted, we pressure cooked, and so we cook the whole seed first, and then so after that we dry the sample and we extract the protein. We want to know during this cooking process with other ingredients as a matrix and how the protein denatured, how the protein respond for the cooking process. So this is the result. And based on this, the first one is protein solubility. Basically, it's one of most important protein function properties. We also determine their denature properties after different cooked. What is the denature protein? How protein being denatured by using DSC thermal properties. Also, the main thing is we want to understand how the secondary structure of a protein changed by using FTIR. Because in the FTIR, so for secondary structure, we have alpha helix, beta sheet. Then so FTIR become very useful tools for determining how this secondary structure change, because we can look at the peak in the is called Amanda 1 in the Amanda 1 is in the 600 to 1700. Then so after this we use if you see like the 1600 to 1700, then so this each one they indicate different the structure. Some like in the has a beta helix, turns beta sheet, alpha helix. So based on this, we can determine how the processing affect the protein structure. So our conclusion for this the work is we found of course after cooking process, protein has been denatured. Because it's denatured, they increase the color. It's become more whiteness when we did the color. But it decreases the protein yield, which means when we extract it, processing the chickpea becomes the protein yield is low. I think if doing the processing, then so they have some interaction protein with other compounds. And also the thing is they increase the water and oil holding capacity, which is good for function property, but decrease the emulsification ability. Meaning for moisture heating, like a pressure cooked, significant change the second structure. So the protein has a very good potential to be used in different food ingredients to make the food products. So based on our results, we published the paper. It's based for different physicochemical function, thermal structural property of isolate chickpea affected by different processing approach. So same thing like you did. I just briefly present the finding we did. But if anyone have interest, want to know more about our funding, please send me email or just tell me so I can send the papers for you to read. And for third step, and so after we isolate the protein, we want to know what other potential application for protein. And recently, in the last couple years, in the last decades, and so one product from protein become very popular is called bipeptides. And so bipeptides actually basically is a protein hydrolase. Is being hydrolyzed, is a protein being hydrolyzed, and to make the short amino acid peptides. So it has a potential health benefit and very active, has a antioxidant, antimicrobial, all these antihypertensive, all these potential health benefits. So usually when make the bipeptides, people use enzymatic hydrolysis because it's a mild condition and has a minimum byproducts, also very specific. We can get the products we want by control different enzyme, by control different the processing condition. So what we did it, we just use the enzymatic method to hydrolyze the protein, the chickpea protein we have. So basically, this is a procedure. This is our chickpea protein. Then so we just we get a mixture. Then so we add different. We have the two enzyme. We add it. I'm going to show the enzyme later on. Then we put it in the water bath for different condition, like for different for the optimal the condition for enzyme. Then we hydrolyze a certain time, cool down centrifuge, get hydrolyzed. So this is basically the hydrolysis condition. We use acrylates and flumazyme. So this we use we use them individually, or we just put a sequential, and to see, because each enzyme they can cut for the protein in the different site. So we just see how they respond, and so how much, and so because acrylates the best the temperature is 450, and so then so pH is 8 is good, but for flumazyme, so it's pH is 7 is good. So that's why we conduct for individual or sequential to see how this enzyme can hydrolyze the protein, chickpea protein, becomes to see any like the bipeptides we can get it. No. So same thing after we hydrolyzed, what we did it, we measure degree of hydrolysis, how much, what extent the protein has been hydrolyzed. Also like protein solubility, and how, because we know like after processed, sometimes the protein solubility reduced. So if hydrolyzed, and after they break down to small amino profile, how the protein solubility. Also, we want to understand how effect of protein, the hydrolyzed processing condition affect the secondary structure and their SDS profile, and also antioxidant, because the main and topic people talk about bipeptides recently said because they have a strong antioxidant properties and has good health benefit. We want to understand how the antioxidant for the bipeptide we hydrolyzed, and so their the properties. So this is our result. And based on our findings, so we found for we measure degree of hydrolysis, then protein solubility. Still same thing. We measure their secondary structure, and so especially, and so we use the software to identify is clearly to show how different like alpha helix, beta sheet, and like how each so for each like area, so we can actually get a percentage for each composition change. We also determine like antioxidant properties by determining by DPPH antioxidant activities. So our conclusion for this work, and still same, this paper has been published like last year. So, but I just briefly talked this one. So it's we found it enzymatic hydrolysis, and they change the second structure. They have more alpha helix show up and the beta sheet and reduced. And the thing also we noticed their increased antioxidant properties and help to improve protein solubilities. So it's really, and we see has potential to use be the food ingredients, but actually is a challenge is being there. The main challenge is right now, if you look at our products, is a mixture, is with all different pipettes, or different peptides, actually is a mixture. So next step is we need to isolate each individual peptides. So that is a challenge for us because you need to use HPLC, all this to do the individual peptides. And the second, so the taste of peptides, because some of the amino acid after hydrolyzed is taste a little bit bitterness. So it's very challenge for people to accept it when you put in the products. So right now, this still is a challenge and for us working on it. So our goal is how we can hydrolyze specific peptides without and also has these all function properties, but still can acceptable the taste. So that's still have a lot of work to do for this. So this paper also and published last year, and so like I said, if anyone interested for the details of work, so I can present this paper for people read it. So last one, I want to briefly talk about our current ongoing research about in this field. Currently, one thing is we're working on different processing, food processing to improve chickpea protein functionality can be used on different food products. So one stuff we did it currently, so we are working on is called high pressure, high hydrostatic pressure for improve function property of chickpeas. Another one is instead of only chickpea protein, and we want to explore more plant-based protein source can either work together with chickpea or has a better function property to be used for food products. So for high pressure processing, so actually I think it's, as a food in the food processing areas, high pressure processing is also called non-thermal technologies. When we say non-thermal, it means they not increase the temperature and just increase the pressure. The pressure can go to the high or ultra high up to 1,000 megapascals. So when I teach students, so sometimes I just tell them, do you know what means like if you have 600 megapascals, what this means? So actually, it's you imagine if you have one disk, like the CD disk before, and think about has 200 elephants. Each elephant is about 3,000 pounds. These all 200 elephants all standing in the one small disk makes the pressure is 600 megapascals, which means the pressure can go very, very high. But good thing is we still can keep in the room temperature, or even sometimes can be in the refrigerator temperatures. So this is fortunately, and so this is our equipment. So we through our USD funding, we secure this equipment. The advantage of these technologies is they can keep, because they're not going to increase the temperature. That's why the food still can be the fresh, taste like fresh. but also can extend shelf life. And also for the HPP, so they can modify the protein structure to improve function properties, such as protein and protein interaction, enhance the gel forming capacity, all these function properties. And that's why in the 2015, and so in the whole world has 100 from the professional in the food engineering, and around the world from industry, for academia, and from government to identify what is the most important food processing in the next 10 decades. Then high pressure processing become the number one. So right now it's become one of most commercial important technologies in the processing field. But the disadvantage, this machine still, the equipment still is so expensive. So the one we have right now is a lab scale, only capacity is 1.5 liters. Just this small size costs about $150,000 to build this one. So that's why right now the challenge for these techniques still is too expensive. So see, okay. So what we did right now, and so we work on the chickpea protein. We work on the chickpea protein, use HPP to processing chickpea protein. After this, we're still working on how HPP techniques affect the protein structure. So especially I'm really interested for their structure, because the structure determines their function properties. And so currently we're working on is we want to understand for first a structure is amino acid profile, how the HPP processing affect amino acid profile. Then second structure like alpha helix, beta sheet. We also determine surface hydrophobicity and the total sulfur content. For this one, actually these two measurements, we actually determine their tertiary structures to see how disulfide bond and how hydrophobicity for the protein changed based on the after we did HPP treatment. Because the structure change going to affect for function property. So we want to compare, okay, after HPP treated, how the function property changed. All this property one. So currently we're still working on this project. So students, also my staff people still working on use HPP treat the sample, then we characterize the function and the structure properties. So last one, I want to talk about another one in which we're working on is the hand-cut protein. Like I mentioned, is ongoing research. It's because except the chickpea protein, we also like to explore more protein source. So hand-cut protein become one topic, and so we are interested in working on that. The main reason because the hemp, so right now become very popular, especially in the United States. And so for hemp, and especially 2018, when the Farmer Bill passage makes the hemp becomes legalized in the US. And so most people interested about oil, especially for CBD. But after CBD after extracted for the oil, the leftover is a hemp protein. So actually, the hemp protein is very promising new alternative plant proteins. I think in the other countries, like in Canada, also other countries in China, already people working on the hemp protein. But in United States, because it's kind of the new protein, and the people still like try to understand how the different varieties, how the growing production side affect the protein the qualities. And so US is especially become the the hemp hard protein in the market in the North America. So hemp protein is good because they have a lot of essential and total amino acid. So it has a kind of like complete amino acid. Even people said their hemp protein, the nutrition profile can compete with like egg protein, and is a highly digestibility. Another important is hypoallergenic, which means you don't need to worry about you're going to get allergy. So it's a mild the flavor. So that's why it's going to be potential for nutrition side. But we did notice the function property is not good because the hemp protein and their each different units is a very compactness. Is they just compacted together. It's really hard to break them down. Also, they present like a covalent disulfide bonds. So that's why the function property show very low. So our job right now, actually our interest, so we just working on the proposal, also the research, the project in the lab to identify, to find it out how the hemp protein and compare to other different other protein. For example, very popular the pea protein, even like soy protein. Also, we want to compare how the protein compare to animal protein like egg protein. And in the meantime, we also want to know what processing technologies can help to improve functionality, to make the hemp not only nutrients, also is functionable, can be used. So right now, what I show it is called, actually is a hemp hard protein. That's why you saw it actually is a white color. And but actually, if you have the skin in there, it's actually show like a tans is a green or gray colors. So also the smell, the taste is not good. That's why I'm sure the hemp protein has a lot of potential, but also has a lot of work going to need to do. That's is all work we want to do. We want to identify this protein. Then we got the database for our for the different plant-based protein. We can see how we can let the protein work together to make the food products. That is our direction for the lab move forward. So by end, I want to send to put the acknowledgement for this last 10 years. So when we work here, so we got the different the funding from like a USDA for local and the local agency. Also, I'm really appreciate has a lot of industry and the collaboration. So they working with us to help us identify some problem, which I really enjoyed. They come us, they tell us what they need. And so we just address, it's a kind of like a question driven. So to help them to solve their issue. Also for all the students, or administration, also so many collaborators work with together. So with that, I want to say thank you very much for your listening and gracias. Any questions, please let me know. Thank you.
Thank you so much, Professor Xu. In this moment, in your screen, we're going to show the question that the participant made, and you can answer each one.
Okay. Could you please talk about amino acid composition of chickpea protein? I mean, replace the protein from animal source. Okay. Actually, this is a good question. And so we did measure chickpea protein and amino acid profile. Actually, we have the one paper and published. So we identify the amino acid profile. Like other all legumes, and so chickpea protein, and also it's not complete protein. They just they have some like it's not it has some essential protein. Essential amino acid is a low content. I think if I remember correctly, I need to check my book that one. I think it's lysine or some has low content. Of course, compared to animal protein mostly, for example, for egg, is complete protein, has all essential protein, or the amino acid. So that's why always we need like to work with the protein with other like cereals, and they can supplement each other for amino acid profile. When you're looking for new source of protein in food industry, what are the most important features you use to check? Okay, that's a good question, honestly. And so when we're looking for new source, and for as a research standpoint, and so what we want to see is if we want to give the recommendation for food industry, we want to them to for its a function good, which means function properties is really important. And when industry make different food products, and so how the protein can be behaved in the products. For example, you make hummus, you make milkshake, all these. So protein has a good emulsification property and solubilities. That's for the academic standpoint. But I'm sure for food industry, they also want to for the cost. You don't want to recommend the protein very expensive. You also want to this protein is easy accessible. Like for example, right now, like a pea protein is already commercialized, and also other protein source is cheaper. But for us, we try to do it. So first, we want the industry to see it's how the function property, especially like I think in most like a water holding and oil holding, emulsification, and the protein solubility. That's a feature we usually check for the protein. How the measure surface hydrophobicity and why important total sulfate content. Okay, that's a good question. Because so like I mentioned, surface hydrophobicity and total sulfate content, when the protein has the structure. So when the amino acid profile is the first structure, the primary structure. Then so alpha helix, beta sheet is the second structure. Then so they have tertiary structure, make the protein can binding together. So this is the bond, disulfide bond, also some hydrophobic bond, bonding them together. So by measure total sulfate content and the surface hydrophobicity, and we know how the like their tertiary structure change. Also important thing is the surface hydrophobicity affect their emulsification properties, also forming capacities. So by measure their like these properties, we can determine their function properties. That's the reason we do that. The digestibility plant-based protein very low compared to animal protein. Within the man, the new source protein have the same nutrition value. Okay, that's a good question actually. And so if you look at the score, and you're right, for the animal protein has a high the digestibility, but for plant-based protein, and that's why we need to do some modified. We need, and especially if you see the score, like if for egg is a 1, different protein has I think if I remember correctly, the score is digestibility is for pea protein, I think about 0.8 some, and for the ham protein we did it is 0.6. And yes, it's compared to the amino protein, it's a little bit low, but like I said, so our goal is can we use a different processing method? We can help to increase digestibility. So in that way, to kind of match the nutrition value for animal protein. But I'm sure not going to be 100%, but still that is our goal to working on that. Thank you for your question. Could you please talk about regulation use hemp? Okay. That's a good point. Actually, because I just did read one proposal recently, so that's why I did some research, and so I can briefly talk about my understanding for regulation. And for hemp, I think it's legalized in the so initially it's legalized in the for research purpose, and to go in the hemp. So that's why like. Initially, like it's only for research base. So in our institute, we are one of 3 institutes in our state can go in hemps. And but I think in the United States, and the first the state legalized hemp is called in the Kentucky. And that's why like we got some hemp protein from one company in Kentucky. Actually, is a company from a person from California. So when hemp legalized in Kentucky, they moved to the Kentucky, and so try to just a lot of farmers growing the hemp. And then so they kind of like after harvest, I think they collect the hemp, make the different hemp products like the oil and the hemp, the proteins. And 2018, I think for the hemp, and the U.S. passed the bill to legalize the hemp in the United States. So right now, for the hemp products, especially for the hemp protein, I think even in the Amazon, even for grocery store, so you can buy the hemp protein. And so even we have the field day for the hemp, and we saw the hemp and protein and even hemp, the flowers, being added in the noodles and for the for demonstration utilization. So I think right now most people still use for like a hemp for CBD. I don't know hemp CBD used for food products or not, but at least for the hemp, the flowers, and the hemp, the proteins, we can use it. Okay. Regarding high pressure processing hemp protein, the trace of oil in the product would have no regular problem. Okay, that's good point. Actually, and so I think that is not an issue because right now, like the hemp protein, actually, and if you check the FDA and the website, the FDA just approved like a hemp protein becomes a grass and just generally considered as a safe. So that's why, and so it no problem if we get a hemp hot protein to do our research or do work. Do you have study effect of green spotting on amino acids? Some people. Okay, actually, this is a good point. Actually, from our research, we didn't do that yet, but I did review some papers, and I realized it's a lot of work people doing this, especially I'm understanding after spotting, and of course amino acids, and so they break down for protein, so make it easy for amino acid going to be the change, but for our own research, we don't do it yet, but it's good direction. Probably for the future, we can do that. But thank you for your suggestion. What characteristic would vegetable protein source have for their industry application approach and genic? What characterizes protein for? Characteristics should animal protein source have for industry application? I want to get clarify it's for industry application, and it means for food or it's a non-food industry application. And for example, we also did some vegetable protein like soy protein and the use for the packaging films. So for that one, we want to have good the strengths, the mechanical properties. And so for the protein, like if for food application, of course, we want to be healthy and so easy digested. So and has a high nutrients. That's for food application. But for industry, so my understanding is for we did some film packaging part. We want to is like it's degradable. Biodegradable also has a good mechanical properties. How about the cost, HP vacation, improve function property of protein is another emerging technologies. I think you raise a good questions because yes, like I mentioned, HPP right now even they have a lot of advantage, but still its cost is expensive, and that is I think a disadvantage use HPP. But actually, if you look at it, the trend, HPP is not the cost is not the higher, and then the people thought. And so even like we have in the United States, especially, and so we have the small farmer, small restaurant owner actually. So they have their own products to make a salad. They want to extend the shelf life. Of course, individual small farm like a restaurant, they cannot have HPP, but so right now in some of the states, they have a cohort. It's some like they bought the HPP machine and they kind of rent, let people go there processing their products. Then you pay like the rent or you pay the fees. So I think it still can be acceptable. Also, I think compared to like before, I think the HPP become more and more acceptable, and for these applications. And other emerging technologies, so that's a good point because most right now people working on the non-thermal and so this one called high pressure processing. Also, some people on the protein they use like the what they call what they call let me think in the words. Homogenization the protein, and also other non-thermal method, and the people use it, but that is for potential. What protein from plant as described as allergenic? And I think in most for the plants, most is a soy protein. If you look at for FDA and described and the 8 source of protein and claim as allergenic, I think one is soy protein, also wheat protein, and that is a 2 protein from plants. I think it's for allergenic. How you work on influence degree of for the protein? Have you worked on the influence? Yes, we did. Actually, for our paper, and so we compare it how like use a different enzyme, and they have different degree of hydrolysis. Because the degree of hydrolysis different, the function properties are different. And if you're interested, I can send the paper or published paper. You read it. We have more details about how this for you to answer your questions.
Thank you so much, Professor Xu, for excellent conference. And thanks everyone for their participation.
You're
this
welcome.
afternoon at the 5 p.m., we will have the conference by Professor Pablo Juliano Collet the high-frequency ultrasound application in extraction and refining process to improve oil recovery. We look forward to everyone to the conference and in October to the PHI International Congress on Research and Innovation in Engineering, Science and Food Technology, IICTA 2021.